Hello. So, very good afternoon to the students who have joined. Please let me know in the chat box if my voice is audible to you, and also let me know if you can see the video. Okay, thank you so much. Thanks for confirming. Now, please let me know if the presentation is visible. The PowerPoint presentation, is it visible to you? Kindly let me know. So you can see the PPT, right? When I'm sharing the PPT, kindly confirm if the presentation is visible or not. Hello, am I audible? We will start shortly when other students will join. I will wait for 4 p.m. dot, then we will start today's session. So please wait till 4 p.m. Please wait, we will start the class at sharply at 4 p.m. So very good afternoon. We will start the session shortly. Kindly wait. Meanwhile, let me know if you can hear my voice and also if you can see the presentation. We will start the session sharply at 4 p.m. And also, due to some network issues and for the smooth running of the class, I have muted you all. But the chat box is open. So please, if you have any query, any question, let me know through the chat box. The chat box is open for you. So if you have to say me, anything, you can let me know through the chat box. Let me know if you are able to hear my voice and also able to see the presentation. We will start the session shortly. Please use the chat box if you have anything you want to say. You can write in the chat box. I'm monitoring the chat box every time. Because of the smooth running of the session, I have muted you all, but the chat box is open. Okay, so if you have to say anything, you can use the chat box. Okay, so it's 4 p.m. I think I should start with the introductions. And meanwhile, when more students will join, they can join the session directly. So, yeah. So, very good afternoon, all. I hope my voice is audible to you and also the presentation is visible to you. Yeah, very good evening. So, let me know if the presentation is visible to you properly. The PowerPoint presentation which I am presenting, I think it is visible to you, right? Okay, okay. Thank you for confirming. So let me start introducing myself. So very good afternoon. I am Mousumi Deb from Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I'm the PMRF fellow from this Department of Chemistry and Chemical Biology, and I am your TA for this NPTEL course of Medicinal Chemistry. So today it is our week one live session. So what we will do, so since it is our first session, so let me go through briefly through some introductions like what we will do in this live sessions. So basically these live sessions are intended for helping you out in the course of medicinal chemistry. So every week, I think on Monday, you get the update of the course content to be uploaded for that particular week. And every week you have a certain portion of this course medicinal chemistry which is uploaded, and then what you have to do is there will be assignments given to you in the NPTEL in the SWAYAM website, which you have to do properly, and you have to submit in a due course of time. So basically, these assignments will be MCQ type of questions, as you can, as I think you have seen already in this website. If not, I will show you at the end of the session also. Okay. So since these questions will be MCQ, so what I will do is in this problem solving session, in this 2 hours of time, I will show you around 15 to 20 MCQ questions. Okay, each question will have 4 options, and in the chat box you have to identify which of the following options will be the correct one. Okay, so before joining the session, in the when the course material is uploaded, you are supposed to cover up the videos, lecture videos, and then you have to try answering out the questions in the chat box. Okay, since it is the practice session, there is no marks given for answering the questions. Okay, so I want all of you to try to answer the questions. Your answer may be right, it may be wrong, but it does not matter. But answering the question is very important. Otherwise, I won't understand that you are understanding the course or not. Okay, so if you have any, one more thing. I am recording this session so that if you have any network disruption in middle, or cannot attend the whole class, so this video will be made available to you, so that you can watch this video, this practice session video, and this presentation, the PowerPoint presentation which I am presenting today, it will also be made available to you, so that you can practice it later also. Okay. So, since I am recording the session, for the smooth recording, I have muted all the microphones of the students. Okay, but the chat box is open here. Always the chat box will be open. So, if you have any doubt, any query, anything, you can let me know through this chat box. Okay, type your query or doubt in the chat box. After each now and then, I am monitoring the chat box. So, if you have any question, anything, I will answer that at any that time. Okay, and also at the end of this particular session, okay, I will share my email ID, so that if you have any problem, any question after the class also, you can just drop an email to me. I will answer your queries through the email. And also there is a discussion forum of this SWAYAM portal, or this particular course of medicinal chemistry. You can post your queries in that forum also. Okay. So I think I should start with the session. If you have any problem or any doubt, you can let me know. Okay, but before starting, let me show you where you have to find this particular medicinal chemistry, this thing. Okay, 1 second. So when you have this kind of content live email, you can directly go to that portion. Just let me show you where to find this particular your course content. Okay, where to find this thing, let me just show you. This problem solving session will be uploaded to you, and in the SWAYAM portal itself. Okay, so just let me do it. Okay, at the end of the session, I also can show you. Yeah. So as you can, I think you can, it is visible to you, right? Is it visible to you? So is it visible to you, right? This particular post. So here you will find there will be another tab will be uploaded. Okay, and that tab you will find this problem solving session updates. There you will find this particular recording, and also you will find that particular PPT which I will be presenting today. Okay. So at the end of the session, I will show you once more if anybody has missed it. Okay, so don't worry. So let me start with the session. So as I have already mentioned, what we will do is I will show you one question. Okay, I will give you 5 minutes of time. There will be 4 options for each question. After you try answering that particular question, I will check your answers, and then we will discuss that particular topic in detail. Okay, I will discuss it in such a way so that if there is any kind of descriptive question in the final examination, you can solve it easily. Okay, so but since in the assignment you have MCQ, so I am putting the MCQs only in this particular. And also there is no marks answering, so if your answer is wrong, you won't be deducted, or you won't be awarded any marks. But participation is very important, so try to answer these questions. So let us see what is the problem number 1. So okay. These things I have already mentioned, but let me just read out one more time. So there will be problem solving session every week. This session will be on every Friday from 4 p.m. to 6 p.m. Okay. Since it is our 12-week course, so we will have 12 problem solving sessions. Okay, on each on consecutive Fridays from 6 p.m. to 4 to sorry from 4 to 6 p.m. And then we will also have a summary session at the end of the course. Okay, these sessions will be live on the Google Meet platform. This is the link of the Google Meet, this link which you are using today. All this is the particular link for the complete semester. Okay, you can save this link or you can write it down at anywhere. So, and you can use this link to join the Friday class every week. So if any network disruption happens, so now this is very important thing. If any network disruption happens from my side, okay, then do not leave the class immediately. Okay, so wait for some time. I will join as soon as possible, but do not leave the session. And if you have any kind of network issue, you can rejoin it anytime the meeting is open. Okay, and since I am recording it and I will share it later, so you won't have any problem. During the session, the students can post their question and doubts through the chat box. If still any doubt persists after class, the students can drop an email to me. I will share my email ID at the end of the session, and I will answer your query through emails also. And this presentation and class recording will be shared to you after class. Okay. So these points I was just wanted to summarize one more. So now let us go to the first question of today's session. So the first, so this is the content of the week one. So the contents are the introduction of the medicinal chemistry, then intermolecular binding forces. We have protein structure and function. Then we have two tutorial. First one is of the acidity, basicity, and related concepts, and the second one is the basic concepts of thermodynamics and kinetics. So these are the topics which we are going to cover up in today's class. Okay. So this is the first question of today's session, which says correct order of a drug discovery program is number one, target discovery. Okay, one second. Hello. My voice is audible, right? Okay. I think my voice is audible. Okay, so this is the first question of today's session. It says that the correct order of drug discovery program is number one, target discovery, number two, lead identification, number three is the lead optimization, number four is hit identification, and number five is preclinical development. So all these are the steps of the drug discovery program. Now you have to identify what is the correct order of all the steps. Your options are number A, 1, followed by 4, then number 2, number 3, and number 5. Your option number B is number 2, then number 1, then number 3, then 4, then 5. Now option number C is number 3, followed by number 4, number 1, number 2, and number 5. And now option number D is number 1, then number 3, number 4, number 2, and number 5. So these are the orders. The what is number one? Number one is the target discovery. Number two is the lead identification. Number three is the lead optimization. Number four is the hit identification. And number five is the preclinical development. Okay. So these are the 1, 2, 3, 4, 5, but it is not in the correct order. So you have to identify what is the correct order of this drug discovery program. Option number A is 1, 4, 2, 3, 5. Option B is 2, 1, 3, 4, 5. Option C is 3, 4, 1, 2, 5. And option D is the 1, 3, 4, 2, 5. I will give you some time. Think and let me know in the chat box A, B, C or D which of the following is the correct answer. So, what is the correct order of a drug discovery program? Let me check if there are any answer in the chat box. Okay, I have found there are several answers. So there is number A, number B. So number A and number B are the two given options till now. Others also try to answer the question. I will give you some more time. So what is the correct order of drug discovery program? Your options are number 1, target discovery, number 2, lead identification, number 3, lead optimization, number 4, hit identification, and number 5 is the preclinical development. You have to find out what is the correct order of all these 5 points. Let me check the chat box now. Okay. So, majorly I have received the answers which are saying that number B is the correct one, but also I have one number A. So, let's check which one of the following is the correct option, A or B. So, the correct answer is it's number A. Okay, so number A is the correct answer, which is first it is the target discovery. Okay, then followed by the hit identification. After the hit identification, we will do the lead identification, then lead optimization, then preclinical development. So it goes like 1, 4, 2, 3, and 5. Okay. So this is the flow diagram of this particularly drug discovery program. So it is a general diagram of drug discovery program. So what is the first thing is, first thing you have to do is the target discovery or the target validation. Okay, so you have to first identify which is your target disease, what is the target of the particular disease. So for example, let us say you are going to discover a drug for cancer. Okay, but for discovering anti-cancer drug, there are several targets or several proteins or enzymes which you can target for a particular anti-cancer drug discovery. So that is the first thing you have to do is first thing you have to identify what is the target going to be the target of your drug discovery program. Then followed by the hit identification. Okay, so hit identification, hit molecules are the molecules first which show some activity as the potential drug molecules, and the best one of this all hit compounds is called the lead molecule. Okay, and this lead molecule is we can get from the scaffold hopping technique, binding mode prediction technique, physical chem property prediction, synthesis planning, in silico admit property also. Then we will have to do this lead optimization. Now the lead molecule, which is the first molecule of the particular series, which is the best one. Then we have to optimize that particular lead molecule. We have to change the functionalities here and there to make it more drug-like. Okay, the drug-like means it has so what is the drug-like properties? All these things we will discuss in our later in this course. Okay. Then we have to do is the preclinical development. In this preclinical development, we have to do the FIH dose prediction and also the translation biomarker discovery and so on. Okay, so this is the particular order of the drug discovery. First is the target discovery and validation, followed by hit identification, lead optimization, and preclinical development. Okay. So, pharmaceutical companies tend to concentrate on developing drugs for the disease which are prevalent in the developed countries and aim to produce compounds with better properties than the existing drugs. A molecular target is chosen which is believed to influence a particular disease when affected by a drug. Greater the selectivity that can be achieved, the less chance of the side effects. A suitable bioassay must be devised which will demonstrate whether a drug has activity against a particular target. Bioassays can be carried out in vitro or in vivo and usually a combination of the tests used. HTS, or it is also known as the high throughput screening, all these things I will discuss in detailing later, so you don't have to worry about it. So HTS, it is called the high throughput screening, involves the miniaturization and automation of the in vitro tests, such as large number of tests can be carried out in short period of time. Compounds can be tested for their affinity to a macromolecular target by NMR spectroscopy. Relaxation times of the ligands are bound to macromolecular shorten when they are unbound. SPR, SPA, ITC are the three virtual methods for detecting whether the ligands bind to the macromolecular targets or not. And virtual screening can be used to identify compounds most likely to be active in the experimental settings. Now, one more thing we have to emphasize on is the lead molecule. So, what is a lead molecule? So, a lead compound is a structure which shows a useful pharmacological activity and can act as a starting point of the drug design. Natural products are rich source of the lead compounds. The agent responsible for the biological activity of a natural extract is known as the active principle. Lead compounds have been isolated from trees, plants, microorganisms, animal venoms, and toxins. A study of medical phosphor indicates that plants and herbs which may contain novel lead compounds. Lead compounds can be found by screening synthetic compounds obtained from combinatorial synthesis and other sources. Existing drugs can also be used as lead compound for the design of a novel structure of the same therapeutic area. Alternatively, the side effects of an existing drug can be enhanced to design a novel drug in a different therapeutic area. So these are the key points you have to remember regarding the drug discovery program and also the lead molecule. Okay. So do you have any doubt regarding the first question? If you have any doubt or any problem, you can let me know through the chat box. Okay. So the first question, if anybody has any doubt, you can tell me now. Also, you can tell me at the end of the session. Okay, fine. Then I will go to the next question. Okay, I think I can go to the next question now. So the problem number 2 is, when a drug is approved for the human testing during the clinical trials, your options are: number A, between phase 3 and phase 4, number B, between phase 1 and phase 2, number C, at the end of phase 4, and number D, at the end of preclinical trials. So when a drug is approved for human testing during the clinical trials. Again, I will give you some time. Think, read the options carefully, and then let me know in the chat box. When a drug is approved for human testing during clinical trials, your options are: number A, between phase 3 and phase 4, number B, between phase 1 and phase 2, number C, at the end of phase 4, and number D, at the end of preclinical trials. So when a drug is approved for the human testing during clinical trials, between phase 3 and phase 4, between phase 1 and phase 2, between at the end of phase 4 and at the end of preclinical trials. Even if your answer is wrong, it does not matter, since we don't have any kind of marking system here. but answering is very important. Okay, otherwise I won't understand that you are following the class or not. And whenever there is any doubt, any query, you can let me know then and there in the chat box. Okay, write your doubts in the chat box. I will try to answer it during the class itself. Okay, let me check the chat box now. Okay, I think I have few answers. Number A, number D, number D, number D, number A. So again, I have two different options as the answers majorly. One is number A and another is number D. So almost okay, I have gotten number B also. Others, do you want to try any other option? Or okay, let me show the answer. Then we can discuss the questions. Okay, so answering is very important. So all of you are answering, which is very good. Okay, now let me check. So the correct answer is it is number D. At the end of the preclinical trials, a particular drug is approved for the human testings. Okay. So this is the clinical trial process. This is a very shortened and very to the point flow chart of the clinical trial process. So as you can see, a timeline of a clinical trial process is around 5 to 15 years. Okay. So special designations can speed up the process. For example, wherever we have any kind of emergency, for example, the COVID-19, we had the emergency. That is why we have to discover the drugs and the vaccines very in a very short time. But in general, a drug discovery process it takes around 5 to 15 years. So first thing is the preclinical trials. Okay, so as you can remember in our previous point, so we have seen one thing. So this is the basic starting of the drug discovery program, which goes into the preclinical development. So after preclinical development, the testing is started. Okay, the clinical trials are started after the preclinical development. So in the preclinical development, what happens is it explores if and how the new drug may work. Okay, then, if the drug is found to be safer or it passes the preclinical trials, the drug is approved for the human testing. Then, the phases of the human testings of the clinical trials are started. So, the phase one is exploring the safety and dosing of the drug. Okay, so the volunteers they actually they come. Some volunteers come forward, and the human testings are they are actually are conducted in the human volunteers. Okay, so first in the first phase, what is explored? That it explored the safety and dosing of the drug. In the second phase, exploring the effectiveness of the drug, and the third phase, exploring the safety and effectiveness compared to the currently available treatment. Okay, so notice these things very crucial, very carefully. That the first step is the first one, phase one, exploring the safety of the dosing of the drug. The phase two is the exploring the effectiveness of the drug. And what is done in the phase 3, exploring the safety and effectiveness together in comparing to all the existing treatment of that particular disease. Okay, when a particular drug is passes through all the phases of the clinical trial, it is submitted for the FDA to the approval. Now, what is FDA? FDA is the Food and Drug Administration. Okay, so it is a government I mean agency of the America, United States of America, and they actually all the drugs. If you have seen, I don't know if you have seen or not properly. So mainly all the drugs they are FDA approved. So it is the highest agency of the drug approval. There are other also in India we have our FSSAI, we have Central Drug Discovery Session also. So all these are the governing agencies. Okay. In UK, there is a particular agency which approves the drugs for the trial. In the Japan also they have their own agencies. So all the agencies they actually approve the drugs. So okay, so when the FDA review happens, they confirm the safety and effectiveness of the drug. So this FDA agency they confirms that yes, this drug is good. After the FDA approval, the drug is released in the market. Okay. After some time, we conduct a phase 4 of clinical trial. So what is the phase 4 of clinical trial? Here we check that if the drug is safe for a long period of time, okay, on the large number of patients. So after, it is also known as the post-market surveillance, okay, to check that after a long time, is if the drug is giving rise to some kind of side effect, or is it affected for, or it is affecting the large number of patients or not. So all these things are monitored in the phase 4 of clinical trial, okay. So these are the phases of clinical trials. Now, what is the preclinical trial? In the last slide also, and here also I have mentioned the preclinical trial, okay, which is basically a bridge between the drug discovery and also the clinical trial process. So what is a preclinical trial? So preclinical trials or non-clinical trials are laboratory tests of a new drug substance or medical devices, usually done on an animal subject, to see whether the treatment really works and if it is safe to test on the humans. The main goals of the preclinical studies are to determine a product's ultimate safety profile. Products may include new medical devices, drugs, gene therapy solutions, etc. So, what is the objective of the preclinical studies? The purpose of the preclinical study is to develop adequate data or to decide that it is reasonably safe to proceed with human trials of the drug. Means a laboratory test of a new drug or a new medical device, usually done on animal subjects, to see if the treatment really works and if it is safe to test on humans. However, main objective of to collect the data to submit to FDA and IND for IND filing. Okay. And then importance of the preclinical trials to determine the dose, toxic dose and pharmacological action, etc. It is the requirement of the regulatory body for performing clinical trials. It is, as ethical point of view, it is necessary to check the safety of the drugs on animals before starting to check on the human being, to check the kinetic profile of the drug and based on it to select the route of administration in the human for the clinical trials. So this point is very important. So in the preclinical trial, we have to identify what should be the mode of administration of this particular drug. So should this drug be given as a tablet or capsule, or should or through by the injection or inhaler? This type of things are they also we find out in the preclinical trials. Okay. So the correct answer of the second question was number D, and I think in the chat box there were several number Ds. Okay. Okay, so I think we should proceed to the next question now. If you have any problem, any doubt, please feel free to ask me through the chat box, okay? Or at the end of the session, you also can ask me your doubts and queries. So next, let's go to the next question. So problem number 3, it says that which of the following lead compound is not derived from the microorganisms? Your options are number A, cyclosporine, number B, curacin A, number C, lipstatin, and number D, rasfonin. Which of the following lead compound is not derived from the microorganisms? If you remember carefully, in the first slide, I have mentioned that lead compounds can be derived from the natural sources like natural products, the trees, the plants, the microorganism, marine sources, toxins, and etc. So here are four lead compounds which are mentioned in the question: cyclosporin, curacin A, lipstatin, and rasfonin. You have to identify which of the following is not derived from the microorganisms. Again, I will give you some time. Try to answer the question. Let me know in the chat box A, B, C, or D which of the following you think is the correct answer of problem number 3. So which of the following lead compound is not derived from microorganisms? Let me know in the chat box A, B, C or D which of the following answers is the correct one. Okay, let me check the chat box now. Okay, I have several answers, which is again very good. Okay, let me check. Number B, number D, number B, number D, number A. So I have three different options as the answer. Number A, number B, and number D. Let me check which of the following is the correct answer. I have got another number B. Okay, let me check which of the following is the correct answer. So the correct answer is it's number B, curacin A. Very good. I have seen several number B's in the chat box. So curacin A is the lead compound which is not derived from the microorganisms. Okay. Now, first, let us understand how this kind of I mean, the lead compound are derived from the different microorganisms. Also, cyclosporin, lipstatin, and rasfonin. These are the lead compounds which are derived from the microorganisms. Okay, so these are the structures. This is the structure of the cyclosporin. This is the lipstatin, and this is the rasfonin. So, microorganisms such as bacteria and fungi, they have provided the rich pickings of the drugs and lead compound. These organisms produce a large variety of antimicrobial agents which have evolved to give the hosts an advantage over their competitors in the microbiological world. This screening of the microorganism became very popular after discovery of the penicillin. The soil and water samples were collected from all around the world in order to study new fungal and bacterial strains, leading to an impressive arsenal of antibacterial agents such as the cephalosporins, tetracyclines, aminoglycosides, rifampicin, chloramphenicol, and vancomycin. So, although most of the drugs derived from the microorganisms are used in the antibacterial therapy, some microbial metabolites have provided lead compounds in the other fields of medicine. For example, aspergillin isolated from Aspergillus aliasis is a novel antagonist of the peptide hormone called the cholecytokinin or CCK, which is involved in the control of the appetite. CCK also acts as a neurotransmitter in the brain and is thought to involve in the panic attacks. Analog of the aspergillin may therefore have potential in treating the anxiety. Other examples of the fungal metabolites are the lovastatin, which was the first clinically useful statins found in the lower cholesterol level, and another fungal metabolite is called the cyclosporin, which is this one, which is used to suppress the immune response after the organ transplants. Lipstatin, this one, is a natural product which is isolated from the streptomycin toxosterone. It inhibits the pancreatic lipase and was the lead compound for the anti-obesity compound orlistat. And finally, fungal metabolite rasponin, isolated from the fungal in the New Zealand, promotes the cell death or apoptosis and of the cancer cell, but not human cells. It represents the potential lead compound in the novel anti-cancer agents. Okay. So now another source are very important for the getting the lead compounds is the marine sources. So in recent years there has been a great interest in finding lead compounds from the marine sources like coral, sponges, fish, and the marine microorganism have a wealth of the biologically potent chemicals and interesting inflammatory, antiviral, anti-cancer therapy. For example, curacin A, which has this structure, is obtained from a marine cyanobacterium and shows potent anti-tumor activity. Other anti-tumor agents derived from the marine sources include the eleutheriobin, bryostatin, dolastatins, and cephalostatins, and helicondrin B. Okay, so in 2010, and simplified analog of helicondrin B was approved for the treatment of breast cancer. Okay, so this curacin A, which is a good anti-tumor agent, is a lead compound derived from the marine sources, whereas the others, which I have mentioned already, are derived from the marine sources. Sorry, some microorganisms. Okay. So the option number B was the correct for this particular problem number 3. Now I will go to the next problem. Problem number 4 says which of the following method can be used for the lead design? Your options are number A IR, number B NMR, number C ESIMS and number D GCMS. Okay. So which one of the following method can be used for the lead design? Your options are number A IR, number B NMR, number C ESIMS and number D is the GCMS. Which one of the following methods we can use for lead design? Let me know in the chat box A, B, C or D which of the following will be the correct answer. Any doubt, any query during the class, you can let me know in the chat box. Okay, so chat box is always open. So if you have any question, you can put it in the chat box. At the end of the session, I will drop my email ID. So any doubt, any query, you can let me know through the email also. Let me check the chat box if there are any answers. Okay, I have several answers, I think, again. Oh, my voice is audible now, or is it breaking still now? Is my voice is fine? I think there is some network issue. Okay, okay, fine. I think there is some network issue. That is why the voice was broken. So let me check the answers are given by you is number B, number B, number C, number B. Okay, number B and C. These are the most given answers till now. So let me check which will be the correct answer for this problem. Okay, so the correct answer for problem number 4 will be, that's number B, NMR. So NMR is the method that can be used for the lead design. Okay. So recently, NMR spectroscopy has been used to design a lead compound rather than discover one. Okay. So discovery and designing are two different things. Discovery and so the design is how we particularly we try and we design a lead compound, and the discovery can be done by serendipity also. Okay. So in essence, okay, let me just first show you the figure. Okay. So what this method is also known as the epitope mapping. Okay. So what is the epitope mapping? And how NMR can help us for designing a particular the lead compound. So in essence, the method sets out the small molecules which are known as the epitopes, which will bind to a specific but different regions of the protein binding site. Okay, so this is a binding, this is a protein, let us say this is a protein particularly, and this is the binding site of the protein. Okay, and then we will have find small molecules or which are known as the epitope. Let's say this one, this blue one. Let's say this is the epitope molecule, and the screening of the first ligand first we will have to do. So the what will happen? These molecules will have no cavity to themselves as they bind to one part of the binding site. But if the larger molecules are designed which links this particular epitopes together, then lead compound may be created which is active and then binds to the whole binding site. Okay, so what is this particular epitope mapping? Now let me say you in detail. Okay, so let us say this is the binding site of the protein, and the particular compound or particular lead molecule have to fit in this particular binding site of the protein. But instead of making a complete whole molecule and then fitting into the binding site, what we can do is we can use small molecules to bind different region of this particular binding site. Let's say first screening in this first ligand, it fits into this position after optimization, and after the second screening, the second epitope or second molecule is fitted into the second position of this protein binding site. Now if we can join this particular two different ligands together, what can happen? We can get a new lead molecule which is able to fit into the binding site of this protein. This method is known as the epitope mapping. What is the example of this particular epitope mapping or particular lead design? So this is the example. So NMR spectroscopy was used in the design of the high affinity ligand for the FK506 binding protein, a protein involved in the suppression of the immune response. Two optimized epitopes A and B. So this is the epitope number A, let us say, which is this one, and we have epitope number B, which is this one. Okay. The two optimized epitope A and B were discovered, which bound to the different regions of the binding site of the protein. So this protein binding site, we have different proteins binding to different positions. Okay, and then what will happen? Two optimized epitopes A and B were discovered, which bound to different regions of binding site, and the structure C. This is the structure C. There was then synthesized where two epitopes were linked by a propyl link. Okay, so basically these two positions are linked with this particular propyl link, and this compound has higher affinity than either of these individual epitopes and represents a lead compound for further development. Okay. So that is how this lead compound or this NMR spectroscopy helps us to identify or to design the lead molecule. So do you understand this particular epitope mapping, or do I need to repeat it? Yeah, I have. It is also useful for designing lead compound, but not as efficient as the NMR. Okay. Yeah, all these questions are from the week one lectures. All these topics which I am showing you is from the week one lectures. Today we will do the week one lectures topics itself. Okay, I will repeat this thing. I will repeat this epitope mapping once more. Don't worry. So basically, whenever we have a protein, so a protein is a this particular, let us say, a enzyme. So enzymes are a protein. Okay, we all know enzyme has a particular active site where the substrate binds, and we have all this particular activity. Receptors are also have active site where the particular active molecule or substrate can bind. So in the lead molecule design, what we can do is instead of designing or synthesizing this molecule directly, okay, sometimes directly designing and synthesizing this kind of molecules are difficult. So what we can do is we can synthesize this, or we can design the lead in parts. Okay, we can break this particular molecule in two halves. Okay, and optimize these two things. So epitopes are the small molecules. We have a protein which have different binding sites. Now we are first optimizing each small molecules in each binding site. So this binding site has a particular this shape. This binding site has particular this shape. So first we will do is we will optimize particular compounds which can fit to the individual portions of the binding site. Then what we will do is we will create a linkage between these two portions of these two epitopes to make a complete molecules which will bind to this particular protein's binding site. This is known as the epitope mapping, and NMR spectroscopy has also helped us in this epitope mapping, which is example is this one. This particular ligand which has this FK506 binding site, okay, and the KD is 0.049 micromolar, which indicates that this compound, this lead molecule has higher affinity than the individual epitopes. Okay, and for designing these things, the NMR spectroscopy helps us. I think it is now little bit clear. When I will share you this particular PPT, you can go through it, and if you have still doubts, I will repeat it in the next class also. Is it fine? When I will share the PPT, you can go through the PPT one time, okay? And I will also share some books also for medicinal chemistry, which will help you to understand the course better. And then after that, if you have still doubt, I will repeat this particular question in the next class. Will it be fine? because today we have to cover more questions. Okay, and also, yeah, you are right. Mass spectra can also help us in the lead molecules, but not as it is not as efficient as the NMR spectroscopy. Okay, so so I think it will be fine then now. If you are not understanding this question properly, go through the PowerPoint presentation once I share this PPT. Then, if it is not clear, I will repeat it in the next class. Okay. Now let us go to the next problem. So problem number 5 says in ADME, A stands for what? Number A, agonism, number B, antagonism, number C, absorption, and number D, affinity. So ADME is a very important term in the medicinal chemistry, a very crucial thing, and we will discuss this ADME in detail later also. But since it is the basic of the medicinal chemistry, I thought to give you a little bit question from this ADME also. So it is very easy. I think all of you can answer this. In ADME, A stands for what? Number A, agonism, number B, antagonism, number C, absorption, and number D, affinity. What does A stands for in ADME? Agonism, antagonism, absorption, affinity. So what is A in ADME? Let me check the chat box now. Okay, I have many options. Absorption, absorption, absorption. I have one A, number C, number C, number C. So majorly I have the answer number C and one number A. Let us check what is the correct answer then. So the correct answer is yes, it is number C, absorption. So what is the other terms of the ADME? So ADME stands for absorption, distribution, distribution, metabolism, and then your yeah then excretion. Okay, so for this particular ADME is a part of the pharmacokinetics. Now, what is pharmacokinetics? So, in medicinal chemistry, two terms are very important. One is the pharmacokinetics, and another is the pharmacodynamics. Okay, so what is the pharmacokinetics? So, in easy language, pharmacokinetics is what our own body is doing to a drug. Okay, what our body is doing to a particular drug, let's say now when we are taking a drug, what our body is doing to the particular drug. Our body is absorbing the drug, then our body is distributing the active components of the drug in all the blood, tissues, and all the active sites. Then our body is metabolizing the drug and excreting the portions which we do not need. So that is known as the ADME and it is a part of the pharmacokinetics. Now, what is pharmacodynamics? Pharmacodynamics means what the particular drug is doing to our body, how the drug is affecting our body, how it is altering the different mechanisms of our body is known as the pharmacodynamics. So these two terms are very important. The one is pharmacokinetics and one is pharmacodynamics. So as we have mentioned already, the ADME, A stands for the absorption. So absorption is the transfer of the drug from its sites of administration to the bloodstream. Most of the drugs are absorbed by the way of the passive transport. Intravenous administration has no absorption, and fraction of administered drug and the rate of the absorption are different. Okay, so absorption is the transfer of the drug. So if you are getting a drug as a capsule or tablet, the drug will be first you have to take the drug and it will go to your stomach and then it is absorbed through the bloodstream. And whenever there is a direct administration, like let's say when you are giving a particular drug through injection, the drug is given directly to your bloodstream, so then there will be no absorption. Okay, so first is the prescription, which gives the patient health. The drug is prescribed, and then the drug is administered by the route and adherence. Then the dose. Then we have this pharmacokinetics, means the bioavailability of the drug, which is the absorption and fast pass metabolism. Then the concentration of drug. It is the distributed to the diffusion, transport, concentration at the target, affinity to the target, molecular effect, and pharmacological effects. All these are the distribution, which is the D of ADME, and this is known as the pharmacodynamics. And then the drug is metabolized, which is M, and then excreted, which is E, which is the clearance. So all terms are very important terms of the medicinal chemistry. The dose, the concentration, the bioavailability, distribution, clearance. Okay, all these are very important points, and which we will discuss later in detail. Okay, so for now, yes, the correct answer was the absorption, which is the number C. So now I will go to the next question. So problem number 6 is, which of the following statements is true regarding the peptide bond formation? So which of the following statements is true regarding peptide bond formation? Now option number A, the peptide bond is not planar in nature. Option number B, the carbon-nitrogen bond is purely single bond in nature. Number C, the cis form is more sterically favored than the trans form. And number D, cis conformation is favorable only when the proline is present. Proline is an amino acid. So you have to now identify A, B, C or D. Which of the following statements regarding peptide bond formation is true? Your options are: number A, the peptide bond is not planar in nature. Number B, the carbon-nitrogen bond is purely a single bond in nature. Number C, the cis form is more sterically favored than the trans form. And number D, cis conformation is favorable only when proline is present. Which of the following statements is true regarding the peptide bond formation? So whenever there is this kind of true false question, the correct incorrect kind of question, read the options very carefully, because maximum students they make mistake during this particular true false kind of questions. Okay. Whenever this type of question or MCQs question, basically this type of detail in detail, so you have to read the options very carefully. So which of the following question is true? Option is true regarding the peptide bond formation. Okay, let me check the chat box now. Okay, I have got many answers. Number C, number A, number C, number B. Uh, Yeah, this particular PPT, okay, the PowerPoint presentation will be made available to you. Okay, so after the class, I will upload, and in the SWAM portal, when you have where you have this particular thing, this particular I mean this particular portion where the assignments are given, there will be a tab of the problem solving session. It will be uploaded soon. There you will find this soft copy of this presentation, and also for the books, I will give you the names of the book at the end of the session. You can download the PDF, and if the PDF is not, you cannot find anywhere, then let me know. I will send you through the email also. Okay. Okay, so I have many answers. I have number C and number A as the answer. So let me check which of the following is the correct answer. So the question was asked that what is the true statement regarding the peptide bond formation? Okay, and the statement which is true regarding the peptide bond formation is it's number D. The cis conformation is favorable only when the proline is present. Okay. So the thing is like that peptide bond is planar in nature. Okay. The carbon nitrogen bond is not purely single bond in nature, rather than it has a single bond double bond component. It is a hybrid kind of in nature, and the cis form is not sterically favored. The trans form of the peptide bond is more sterically favored than the cis form. So this is the peptide bond. Okay, we have in the peptide bond. This is the primary structure of the protein. So amino acids, they are joined together by through a amide linkage, which is known as the peptide bond. So in this peptide bond, this is pure. This is planar in nature. Okay, and this particular peptide bond is okay. Let me just show you. So this is the planar in nature, because and then why this is planar in nature? Because this nitrogen, a lone pair of nitrogen and the lone pair of oxygen, they stay in the resonance, this kind of plus minus resonance, which resonance give this particular bond, this CN bond, the double bond nature, and which makes this particular peptide bond planar. Okay, and this is that the trans form is more favorable sterically, because this oxygen and nitrogen, they are opposite sides, so this is favored conformation of the peptide, which is the trans form, and the steric clash will be happen if there is a cis conformation. That is why the cis conformation is not favored. But when there is proline molecule present, so peptide bond of the proteins is planar in nature as the result of the resonance of the structures, which is seen in this figure number 2.3, and this gives the peptide bond a significant double bond character, which prevents the rotation. As a result, bond rotation in the protein backbone is only possible for the bonds on either side of the peptide bond. This is an important consequence for the protein tertiary structure. So there are two possible conformation of the peptide bond. The trans conformation is the one that is normally present in the protein, and the cis conformation leads to the steric clash between the residues. However, the cis conformation is possible for the peptide bonds next to a proline residue. So the proline is an amino acid which is not, this is a cyclic amino acid. That is why whenever there is a proline present, then the cis conformation will be stable. Otherwise, the trans conformation is just stable for the peptide linkage. Okay. So option number D was the correct one. Now I will go to the next problem. So problem number 7 says, what are the primary stabilizing forces of the alpha helix and beta sheet structure? Your options are number A, hydrophobic interaction, number B, hydrogen bonding interaction, number C, covalent interaction, and number D, disulfide bond. So which of the following is the correct answer? Which of the following is the primary stabilizing force of the alpha helix and beta sheet structure? So from the question, it seems like there will be multiple correct answer, but there will be only one correct answer. Okay, I will correct it later on. The question should be like this: what is the primary stabilizing force of the alpha helix and beta pleated structure? Okay. Number A, hydrophobic interaction. Number B, hydrogen bonding interaction. Number C, covalent interaction. And number D, disulfide bond. So, what are the primary stabilizing forces of alpha helix and beta pleated sheet structure? Hydrophobic interaction, hydrogen bonding interaction, covalent interaction, or disulfide bond interaction? Let me check the chat box. Okay, number B, number A, number B, number A. So I think there are number A and number B. There are conflict between these two options. Let me check which of the following is the correct option, number A or number B. So the correct answer is it is number B. Hydrogen bonding interaction is the primary stabilizing force of the both alpha helix and beta pleated sheet structure. Okay. So the secondary structure of the protein consists of the regions of ordered structure adopted by the protein chain. Okay, 1 second. In structural proteins such as wool and silk, secondary structures are extensive and determine the overall shape and properties of such protein. However, there are also regions of secondary structure at the most of the proteins. There are three main secondary structure: alpha helix, alpha beta pleated sheets, and beta turn. So alpha helix results from the coiling of the protein chain such that the peptide bonds making up the backbone are able to form the hydrogen bonds between each other. These hydrogen bonds are directed along with the axis of the helix as shown in this figure. The side chains of the component amino acid stuck out at the right angles from the helix, thus minimizing the steric interaction and the further stabilizing the structure. Other less common type of helixes can occur in protein, such as the 310 helix, which are the more stretched than ideal alpha helix, and pi helix, which is more compact and extremely rare. Okay, so here you can see there is a hydrogen bonding between the carbonyl oxygen and the hydrogen of the NH of the next, the next chain, and that is how this particular hydrogen bonding stabilizes the alpha helix. And we also have a beta pleated sheet and beta turn also. So beta pleated sheets is the layering of the protein chains in one top on the other, which is this one. And here too the structure is held together by hydrogen bonds between the peptide chain. The side chains are situated at the right angles to the sheet. Okay, the side chains are situated either over or the below the plane of the sheet. And once again, there is due to the steric interaction. The chains of the beta sheets can run in the opposite direction or anti-parallel or the same direction in the parallel direction. And also we have this beta turn, which in showing the hydrogen bonding between the first and third peptide bond. Okay, which is known as the beta turn, which is extremely, it is not of the majorly it is the alpha helix and the beta pleated sheet. Okay, so hydrogen bonding interaction was the correct answer. Now problem number B says the fibrous and globular proteins are associated with dash and dash structure of the proteins respectively. Your options are number A secondary and primary, number B primary and tertiary, number C secondary and tertiary, and number D tertiary and secondary. So fibrous and globular proteins are associated with which structures of the proteins? Secondary primary, primary tertiary, secondary tertiary, or tertiary secondary? The fibrous and globular proteins are associated with which structures of proteins? What will be the correct answer? Which option will be the correct answer? The fibrous and globular proteins are associated with which structures of the protein? Fibrous and globular proteins are associated with which structure? Let me check the chat box, or I will give you 1 more minute. Okay, let me check the chat box now. So I have several answers. number D, number D, number A, number C, number B. So I have gotten A, B, C, and D, four options together in for this question as answer. So let me check which of the following will be the correct answer. So the correct answer, the fibrous and globular proteins are associated with the secondary and tertiary structures of the protein respectively. Okay, number C is the correct answer. The fibrous proteins are associated with the secondary structure and the globular proteins are associated with the tertiary structure. So fibrous proteins, when the amino acids are joined in the straight chain and make a fibrous and sheet-like structure, it is known as a fibrous protein. And when a polypeptide chain of amino acids became folded and makes a globular structure or ring or spherical, it is known as a globular protein. So fibrous protein is a class of insoluble proteins such which constitute the main structural elements of our body. And the globular proteins are the protein which is soluble in water and tend to be involved in the metabolic function. Fibrous protein has long and narrow shape and have a helical or sheet-like structure, whereas the globular protein has a round and spherical shape and have a folded ball-like structure. The fibrous protein in the secondary structure it is associated with the secondary structure, and this globular protein is associated with the tertiary structure of protein. Fibrous protein consists of the repetitive amino acid sequence and it is insoluble in water, acid, and bases. Whereas the globular proteins consist of an irregular amino acid sequence and soluble in water, acid, and bases. Fibrous protein have strong intermolecular interaction, whereas the globular protein have weaker intermolecular interactions. Fibrous proteins are less sensitive to the changes of temperature and pH, but the globular proteins are sensitive to the changes in the temperature and pH. Fibrous proteins are involved in the forming structures like as collagen, elastin, actin, myosin, fibrin, and keratin. And the globular proteins are involved in metabolic reaction like enzyme, hormone, hemoglobin, immunoglobulin, all the examples of the globular protein. The fibrous proteins are basically the skeletal proteins like the collagen of our skin, the keratin of our hair, all are this example of the fibrous protein, whereas the enzymes, hormones, all the hemoglobin, these are the examples of the globular protein. Okay. So option number C was the correct one. Now quickly I will go to the next problem. So problem number 9 says identify the correct statement. Number A, in the interior of a globular protein, histidine will be present. Number B, valine will stay in the interior of a globular protein. Number C, serine is in a non-polar amino acid. And number D, alanine is a hydrophilic amino acid. So you have to identify which of the following statement is correct. Again, I will repeat the options. Number A, it says in the interior of a globular proteins, histidine will be present. Number B, valine will stay in the interior of a globular protein. Number C, serine is a non-polar amino acid. Number D, alanine is a hydrophilic amino acid. So which of the following statement is the correct one? You have to identify. Read the options carefully and then let me know in the chat box. What do you think? A, B, C or D, which will be the correct answer? Number A, in the interior of globular proteins, histidine will be present. Number B, valine will stay in the interior of globular protein. Number C, serine is a non-polar amino acid. And number D, alanine is a hydrophilic amino acid. Let me check the chat box now. Okay, I have few answers. Number C, number A, number B. So A, B, and C. I have three different options for the answer of problem number 9, which I have to now check which of the following will be the correct one. So the correct answer for problem number 9 is, it is number B. Valine will stay in the interior of the globular protein. This statement is the correct one. First, let us see the classification of the amino acids. So we have non-polar amino acids which are considered as the hydrophobic. Okay, so this amino acid does not have any kind of polar side chain. So the examples are the amino acids which are non-polar is the glycine, alanine, valine, cysteine, proline, leucine, isoleucine, methionine, tryptophan, and phenylalanine. So whenever you are reading the amino acid, try to remember the three-letter code like GLY for glycine, and also the one-letter code which is the G for glycine, A for alanine, and all those things. Okay. So these 10 amino acids, these are the non-polar or hydrophobic. Then we have the hydrophilic amino acids or the polar amino acids. So polar amino acids can be of two types. One can be the hydrophilic amino acids can be polar, I mean which is the non-charged like serine, threonine, tyrosine, asparagine, glutamine. These are polar but these are not charged. And we have the charged amino acid which are also hydrophilic. The positively charged amino acids are the lysine, arginine, or histidine. These are also known as the basic amino acids. And the negatively charged amino acids are the aspartic acid and glutamic acids, which are also known as the acidic amino acids. Okay. Now, globular protein, which I have discussed in the last slide also. So, these are named for their approximately spherical shapes and are most abundant proteins in the nature. The globular proteins exist in the enormous variety of the three-dimensional structure. Nearly all globular proteins contain the substantial number of alpha helices and beta sheets folded in a compact structure stabilized by both polar and non-polar interaction. One second. Yeah. The globular three-dimensional structure forms spontaneously and maintained as a result of the interaction among the side chain of the amino acids. So this is important. Okay, the spherical shape of the globular protein is basically due to the interaction between the side chains of the amino acid. Okay, so most often hydrophobic amino acids. Okay, hydrophobic amino acids mean the non-polar amino acids like valine, glycine, alanine, cysteine, proline, isoleucine, and so on. They are buried and closely packed in the interior of the globular protein out of the contact of the water. Okay, so non-polar amino acid, they stay in the interior of the particular globular protein. They are packed inside the globular protein. And the hydrophilic amino acids, this can be charged, that means the charged or uncharged or polar amino acids. They lie on the surface of the particular globular proteins, and these are the exposed into the water. Okay, consequently these globular proteins are the very much water soluble, and which actually required for the activity of the different kind of enzymes or hormones also. So this diversity of the protein structure reflects the remarkable variety of function performed by the globular protein like binding, catalysis, regulation, transport, immunity, cellular signaling, and so on. Okay, so all the options, if you remember, if you see the options, the first one says the interior of the globular protein histidine will be present, which is false because histidine is a hydrophilic amino acid. It will stay in the exterior of the globular protein. Valine will stay in the interior, which is correct because valine is hydrophobic. Serine is non-polar. This is also incorrect because serine is a polar amino acid. And alanine is a hydrophilic amino acid. This is also false because alanine is a hydrophobic amino acid. Okay, that is why option number B is the correct one. So, okay, this was the problem number 9. Now I will go to the problem number 10, which says which of the following statements is false for the determination of the 3D structure of the proteins. Number A, best way is to crystallize a protein. Number B, NMR can also provide the static conformation of the proteins. Number C, the cryo-electron microscopy can directly show the image, no need for the crystallization. And number D, for NMR, highly concentrated solution are needed. So, which of the following statements is false for the determination of the 3D structure of protein? You have to identify the statement which is incorrect for the among the following options. Okay. For the among statement which is incorrect regarding the 3D determination of the proteins. Your options are number A. The first statement says the best way is to crystallize a protein. Option number B says NMR can also provide the static conformation of the protein. Number C, it says the cryo-electron microscope can directly show the image, no need for the crystallization. Number D, for NMR, highly concentrated solutions are required. Which of the following statements is false? Identify the statement which is not correct. Let me check the chat box because we don't have much time. Okay. Number D, number D, number D, number D. Number B is right. Okay. I think you want to say that number B is the correct answer. Okay. So maximum I have got the number D and one number B as the answer of this question. Okay. Let me check which of the following is the correct answer. So the correct answer is, or the statement which is false will be, it is number B. NMR can also provide the static conformation of protein. This statement is not correct. This statement is false. Why? First, let us discuss about the 3D structure of the why 3D structure are necessary. So the 3D structure of the proteins are the precious sources for the information, like shape and domain structure, protein classification, prediction of the function of the uncharacterized protein, interaction of other macro with the other macromolecules, interactions with small ligands, metal ions, nucleotides, substrates, cofactors, and inhibitors, evidence for the enzyme mechanism, structure-based drug development, post-translational modification like disulfide bond and N-glycosylation, experimental evidences for the transmembrane domain. So these are the precious informations which we can get from the 3D structure of protein. So now regarding the determination of the 3D structure of protein, there are different techniques. To determine the 3D structure of proteins, one best way is the X-ray crystallography. If you can isolate a crystal structure of the protein, that is the ultimate structural evidence. Methods of determining the complete 3D structure usually are of three types. Okay, one is the X-ray crystallography, which is the most reliable one. Then we have the NMR spectroscopy. Very high field of NMR machine is required. Through NMR spectroscopy, you can determine the 3D structure of protein. However, there are some disadvantages of the NMR spectroscopy. You cannot handle very large molecules in the NMR, but at the same time, there are advantages as well. In X-ray crystallography, if you cannot crystallize the protein, you cannot get the crystal structure. The crystallography needs a crystal, the structure is the static one. We get an advantage structure of the crystal. But in NMR spectroscopy, it is recorded in the solution. I don't know how many of you are familiar with the NMR spectroscopy. We will discuss it in detail in the our third or fourth week course content. I think there is a NMR spectroscopy also. So NMR spectroscopy is recorded in the solution, and so you can see the exactly dynamics of the system. But you need a highly concentrated solution to get the signals. The peaks are actually very complicated one, and you should have isotopically labeled carbon, isotopically labeled nitrogen, and the technique to do that. By the NMR, you can do the structural analysis, but not very large proteins. Proteins, there is a limit. For 60 kilodalton, you can do the structural elucidation. Okay, so for the 3D structure, the best way is to do is the crystallization of the protein. Okay, the NMR cannot give us the static conformation because crystallization is like for the freezing the particular conformation of the protein. But the NMR, we record the NMR in the solution. So in the solution, the protein will be in the dynamic motion, not in the static motion. That is why NMR cannot give us the static conformation of the protein. So problem number B is the incorrect one. Cryo-electron microscope can also directly show the image. And for NMR, if you want to use the NMR for the 3D structure determination of the protein, you have to use very high concentrated solutions and isotopically labeled carbons and nitrogens for the particular structure elucidation. Okay, so option number B was the false one. Now problem number 11, it says that cisapride, a gastric agent, was withdrawn from the market because number A, it lowers the iron absorption of the body. Number B, it has poor bioavailability. Number C, it inhibits the HERG potassium ion channels. And number D, it invades the blood-brain barrier. So cisapride, a gastric agent which is withdrawn from the market, and what was the reason? You have to identify the correct reason. Your options are number A, it lowers the iron absorption of the body. Number B, it has poor bioavailability. Number C, it inhibits the HERG potassium ion channels. And number D, it invades the blood-brain barrier. Identify the correct answer. Why Cisapride was withdrawn from the market? What is the reason? A, B, C, or D? Let me check the chat box. Okay, number C, number D, number D, number A, number C. So number C and number D are the most given answer as I can see it. Okay, let me check which of the following will be the correct answer. So the correct answer will be, it is number C. It inhibits the HERG potassium ion channels. So this is the structure of the cisapride. Okay, so this is an example of the early tests of the potential toxicity. So in vivo and in vitro tests are often carried out in an early stage to find out whether the lead compound or the candidate drugs are likely to have any certain type of toxicity. One such test is to see whether the compounds inhibit the HERG potassium ion channels in the heart. This HGRT gene, it codes for this protein which is called with the so-called human ether agogo related gene, and several promising drugs are withdrawn from the very late stage of their development because they were found to inhibit this HERG potassium ion channels. Inhibition can result in the disruption in the normal rhythm of the heart, leading to the fibrillation, heart failure, and death. Gastric agent cisapride and antihistamine terfenite had an gastric agent cisapride and the antihistamine drug terfenadine both had to be withdrawn from the market because of this problem. A large variety of the structures have been found to have this unwanted effect, and so tests to detect the property best done as early as possible in order to remove this property as a part of the drug optimization process. Okay, so option number C was the correct one. Now let me go to the next problem. So problem number 12 says the term used to signify the biological effect of a preparation without the active drug that appears to induce a beneficial effect similar to that of the active drug preparation is known as. Your options are number A placebo, number B an agonist, number C me too drug, and number D is the peptidomimetic. The term used to signify the biological effect of a preparation without the active drug that appears to induce the beneficial effect similar to that of the active drug present in the preparation. What it is called? Your options are number A placebo, number B agonist, number C me too drug, and number D peptidomimetic. Read the question carefully and then answer the question. So, what is the term used to signify this particular type of compound? Let me check the chat box. Okay, so if anybody has any question, okay, you can let me know in the chat box. Number A, number A. So I think most of the answers are given is number A. Okay, let me check if number A is the correct answer. So the correct answer will be, yes, it is number A, placebo. So what is a placebo? A placebo is defined as a substance sorry, that has no known specific pharmacological activity against the condition being treated. Placebos can be therapeutically beneficial to some patients when they give rise to a placebo effect. So what is a placebo effect? A fake treatment can make someone better simply because they expect it to help. Okay, so it can, so a compound, it can be a real drug or a chocolate also. Okay, let me just it is a similar thing. So if somebody tells you that this is a drug, it will make you feel better, and if the particular compound, for taking the particular compound, if the patient, if they feel better, as long as they believe it will help. This is known as the placebo effect. Okay. So placebo, as I have mentioned, it is defined as the substance that has no known pharmacological activity, but is beneficial to the patient because they know this will help them, and it will induce the placebo effect. So what is agonist? So I have mentioned here. So let me just tell you, agonist is the drug that occupy the receptors and activate them. Antagonists are the drugs that occupy the receptor but do not activate them. Antagonists block the receptors activation by the agonist. And we have also, so this is the agonist and antagonist. Then we have the Me Too drug. Me Too drug is a drug that is structurally very familiar to already known drug with only one minor differences. The term Me Too carries a negative connotation. However, Me Too products may create a competition and drive the prices down. Okay, Me Too drugs can be broadly defined as the chemically related prototype or other chemical compound which has an identical mechanism of action. Also, we have the peptidomimetic. What is a peptidomimetic? These are the molecules which significantly reduced peptide nature that mimic the bioactive conformation of peptide, thus retain the ability to interact with the biological target and cause some biological effect. Small protein-like chains are designed to mimic the peptide, arise either from the modification of existing peptide or by designing the similar system. For example, peptides or beta peptides. Okay, so these are the terms which we have used for this particular question. Now, I will go to the next problem. Problem number 13 says, which country is world's largest producer of the generic drugs? Your options are number A, USA, number B, China, number C, India, and number D, Japan. Which country is world's largest producers of the generic drugs? USA, China, India, or Japan? So, which country is world's largest producer of generic drugs? Let me check the chat box if there are any answers. Okay, I have few answers. Number C, number D, number C, number C. Okay. No, homeopathy do not work on the principle of the placebo effect. So homeopathy is, there is a particular active agent in the homeopathy. Okay, that component which are given by the homeopathy medicine practitioners, they give you a compound which is which affects actually your health. It affects. Okay, it works. But placebo is not a particular compound which is a drug. So basically, somebody can give you a powdered sugar and they can tell you, okay, this is a medicine, take it and you will feel better. If taking that particular powdered sugar, you feel better immediately for a particular random your problem, then it is a placebo effect. But in case of homeopathy, they give some active components which acts on our body's mechanism. Okay, so homeopathy and placebo effects are not connected. Okay, so maximum I have the answer C and one answer D for this question number, I think 13. So let me check if it is correct or not. So, the world's largest producer of generic drug? Yes, indeed, it is number C. It is the India is the world's largest producer of the generic drugs. So, a generic drugs or is a drug which is produced or distributed without the patent protection. The generic drug may still have a patent on the formulation but not on the active ingredient. A generic must contain same active ingredient as the original formulation. When the generic products became available, the market competition often leads to the substantially lower prices for both the original brand product and the generic forms. In US, the drug patents give the 20 years of protection, but they are applied before the clinical trials begin. So, effective life of the drug patent leads to the tends to between 7 to 12 years. Okay, so all this patenting things, I think at the it is not directly involved in our medicinal chemistry course, but if you have interest, I can give you a brief about this all these topics, but at the end of our course, okay, and the summary sessions. I will give the brief about this all these patent things and all this. So India is world's largest manufacturer of the generic medicines. All the companies, pharmaceutical companies like Sun Pharma, Dr. Reddy's, Cipla, Lupin, Zydus, Aurobindo Chemicals, then Glenmark, Torrent Pharma, Ranbaxy, Natco, all they are producer of the generic medicines. Okay. Now I will go to the next problem. Problem number 14 says which of the following pairs does not represent an acid and its conjugate base? Your options are HNO3 and NO3 minus. Option B is H2SO4 and SO4 2 minus. Number C is the HSO4 and SO4 2 minus. And number D is H2SO4 and HSO4 minus. Which of the following pairs does not represent an acid and its conjugate base? A, B, C, or D, which of the following is the correct answer? Let me know in the chat box. Let me check the chat box. So, what are the correct answer? Number D, number B. But how so much diluted medicine works in the body? So, in case of homeopathy, the dilution actually there is a dilution effect. Yes, based on the dilution effect. Okay, I will give you a brief about this diluted dilution method and the homeopathy in our summary session because it is not directly involved in our course. But since you have the interest in this, I will. I have to also, I mean, make some things so that you understand it better. So, will it be fine if I give you a brief about this homeopathy medicine and this diluted things in the last session in our summary sessions? Will it be fine? Or our next session also, if I have time, I will give you a brief about it. Okay. Homeopathy medicine actually these are based on many natural products. I mean the things which are availed from the nature. These things are affected. That's why because of this dilution effect, the homeopathy takes longer time to affect in the body. But since it is the natural product type of things or the naturally availed things, what happens because of the homeopathy there are lesser side effects. which is the more side effect in the allopathic, which are the chemical things. Okay. So next session I will try to give you a brief about it. Otherwise, I will try to make a PPT or the presentation over it and give it in the last session, the summary session. Okay. So I have answers like B and C and D also. Okay, let me check which of the following is the correct answer. So the correct answer will be, it is number B. H2SO4 and SO4 2 minus, these are not acid, conjugate acid and conjugate base. So in a very easy language, in the Bronsted-Lowry acid-base theory, a Bronsted acid and Bronsted a base. Okay, so what is the difference between a conjugate acid and conjugate base is the proton. As you can see, HNO3, NO2, NO3 and NO3 minus, there is a less proton here. Here there are two protons lesser, so that is why this pair is not an acid base pair. But here also there is one proton less, and this also have one proton less. Okay, so basically the conjugate acid base pair, they will have a difference of one proton molecule, one proton, one H plus. The difference of one H plus will be there. So these are the different acids and their conjugate base. This is perchloric acid and perchlorate ion, H2SO. So these are the conjugate acid base pairs. Okay, H2SO4, HSO4 minus, HNO3, NO3 minus, H3O plus, water. Okay, so all these are the conjugate acids and the acids and their conjugate base, and all will have a difference of one proton. Okay, so remember, conjugate acid base pair, they have a difference of a proton. Now problem number 15, it says what will be the correct order of the acidity strength? Your options are number A, Hi greater than HBr, then HCl, then HF. Then number B is the HBr greater than HF, greater than HCl, greater than HI. Number C is the HF greater than HCl, greater than HBr, then HI. And number D is the HCl, then HI, then HBr, then HF. So what will be the correct order of the acidity strength of this particular halogen acids? Okay. Let me check the chat box. Number A, number C, number C, number C. So I have number C and number A, two different options. Okay, well, where to find this PPT, all this thing I will answer at the end of the session. Okay, so stay till the end. I will share it at the end of. At the end, I will summarize where to find all these things, so don't worry. So the correct answer will be, it is number A. HI, then HBr, then HCl, and then HF. Okay. So this is the particular ratio thing. Okay, the stronger the the strongest acid is the HI, then HBr, then HCl, and HF. And if you ask for the base, the strongest base is the fluoride ion, then chloride ion, then bromide, then iodide. Okay. So this is the bond strength, okay, and bond dissociation energy and the pKa values. So as you can see, we all know the lesser the pK, the more stronger the acid is. Okay, so down the group, whenever so all this fluorine, chlorine, bromine, and iodine are in the same group. So down the group, the bond strength decreases and the bond length and acidity increases. Larger anion are stabilized by the greater charge dispersion across the period from right. The electronegativity and acidity increase, and anion is stabilized by more highly charged nucleus. Okay, so what actually happens, like HF, it has a very small bond length. Okay, that is why the bond dissociation energy is very high, which is 136 kilocalorie per mole, whereas for the HI, where the bond length is greater, the bond dissociation energy is less. Okay, that is why dissociation of this HI bond is much easier than the HF bond. So if this bond is broken easily, the more protons will be generated and more acidity will be more. That is why HI is the most strongest acid, followed by HBr, then HCl, then HF. Okay, so the correct order is number A. Now I will go to the next problem. So problem number 16 says, what is the reason of the lower pKa value of the phenol than the cyclohexanol? Your options are: number A, cyclohexanol is more thermally stable. Number B, pi electron cloud of the aromatic ring takes place in delocalization. Number C, the specific reason cannot be determined experimentally. And number D, cyclohexanol has more chances of hydrogen bond formation. What is the reason of the lower pKa value of the phenol than cyclohexanol? Your options are: number A, cyclohexanol is more thermally stable. Number B, pi electron cloud of the aromatic ring takes part in the delocalization. Number C, the specific reason cannot be determined experimentally. And number D, cyclohexanol has more chances of hydrogen bond formation. What is the correct reason of the lower pKa value of phenol? Read the options carefully and then let me know in the chat box, A, B, C or D, which of the following will be the correct answer. So what is the reason for the lower pKa value of phenol than cyclohexanol? Your options are: number A, cyclohexanol is more thermally stable. Number B, pi electron cloud of aromatic ring takes place in delocalization. Number C, specific reason cannot be determined experimentally. And number D, cyclohexanol has more chances of hydrogen bond formation. Let me check the chat box. Okay, number B. I think number B is the only given answer for this question. Okay, let me check if number B is the correct answer or not. So the correct answer is yes, it is number B, pi electron cloud of the aromatic ring. In case of phenol, it takes place in the delocalization. So basically, what happens? Even the delocalization into the hydrocarbon part of the molecule increases the acid strength. So lower the pK value of phenol means phenol is more acidic than the cyclohexanol. Okay, so why phenol is more acidic than cyclohexanol? Because of the delocalization. Okay, in the phenol, the phenolic OH group is directly attached with the benzene ring. Okay, so this on deprotonation, the negative charge is again on the negative charge will be delocalized and not on the oxygen atom. But not only on the oxygen atom but into the aromatic ring itself. The effect of this stabilize the phenoxide anion relative to the conjugate base of the cyclohexanol, where there is no delocalization possible. This is reflected in the pK values of the two compounds. Okay, the 10 for phenol and the 16 for cyclohexanol. So as you can see here, the phenoxide anion, which is the conjugate base of the phenol, here the negative charge is delocalized into the aromatic ring, which this lone pair of the sp2 orbitals do not overlap with the pi electron system. Okay, and this lone pair, this negative charge can travel throughout the ring, which increases the stability of the conjugate base. But then that is why the pK value of phenol is lower than the cyclohexanol, where means phenol is more acidic than the cyclohexanol. Okay, so option number B was the correct one. Now problem number 17 says which of the following statements is correct? Number A, piperidine is a stronger base than pyridine. Number B, pyridine is a stronger base than piperidine. Number C, both have the similar basicity. Number D, the basicity of these two molecules depends on the solvent. Which of the following statements is correct? This is the structure of pyridine, this is the structure of piperidine. Okay. So number A, piperidine is a stronger base than pyridine. Number B is the pyridine is a stronger base than piperidine. Number C, both have the similar basicity. And number D, the basicity of these two molecules depends on the solvent. Which of the following statement is correct? Option number A, piperidine is a stronger base than pyridine. Option number B, pyridine is a stronger base than piperidine. Number C, both have similar basicity. And number D, basicity of these two molecules depends on the solvent. Let me check your answers now. Number A, number A. So I have majorly gotten, okay, I have got number A, number B also as answer, and number D also as one answer. Okay, let me check which of the following will be the correct answer. So the correct answer will be number A. Piperidine is a stronger base than pyridine. So pK associated with the protonation of the piperidine, a typical secondary amine, is about 13. The equivalent pK of the protonation of pyridine, a compound which has similar heterocyclic structure, but the lone pair of the particular nitrogen is in the sp2 hybrid orbital rather than the sp3 hybrid orbital of this particular piperidine. Okay, so in case of the piperidine, the pK value is 13, and in the pyridine, the pK value is 5.5. Pyridine is a weaker base than the piperidine, and its conjugate acid is a stronger acid. Nitrile, whose lone pair is in the sp hybridize, are not basic at all. The lone pairs with the more p character, like for sp3 orbital, there is 3/4 of the p character, and in the sp orbital, there is a half of p character, are the higher of the energy. They spend more time further from the nucleus and therefore are more basic. Okay, so higher, more the p character, more is the basicity. You have to remember. Okay, so that is why piperidine is a stronger base, because here the lone pair is in the sp3 hybrid orbital, whereas the in the case of pyridine, the lone pair is in the sp2 hybrid orbital or hybridized orbital, which has the lower energy. Okay, so that is why piperidine is a stronger base than the pyridine. So option number A was the correct answer. Now, problem number 18, it says that identify the incorrect statement. Your options are: number A, DMF shows two NMR signals for methyl proton at the room temperature. Number B, DMF shows the single NMR signal for the methyl protons at room temperature. Number C, DMF shows a single NMR signal for the methyl proton at the higher temperature. And number D, the NMR of DMS shows the time average signal in the higher temperature. Which of the following statement is incorrect? I will give you some time. Read the options carefully and then let me know which of the following statement is the incorrect statement. Which of the following statement is incorrect? Number A, DMF shows two NMR signals for the methyl proton at room temperature. Number B, DMF shows a single NMR signal for the methyl proton at room temperature. Number C, DMF shows a single NMR signal for the methyl proton at high temperature. And number D, the NMR of DMF shows a time averaged signal at the high temperature. Let me check the chat box now. Okay, I have answer like number A, I have one number B also. Okay. Majorly you think it is either number A or number B. So let me check which of the following is the correct answer. So the statement which is incorrect is, it is number B. Okay. DMF shows a single NMR signal for the methyl proton at room temperature. This statement is incorrect. Why? Let me show you. So, okay. Yeah. So the NMR spectrum of the N,N-dimethyl formamide or DMF is recorded around the room temperature. Then it will show two signal for the methyl groups. Okay, although it might have been unexpected that the two methyl groups would be magnetically equivalent environments, these two methyl groups. Okay, the dimethyl formamide is represented as the resonance structure of the two form. One is this one and another is this one. So the resonance between this particular nitrogen lone pair and oxygen, it gives rise to the double bond character of the CN bond, which makes the DMF, this particular DMF, double bond, give this particular bond a double bond character, and thus there is a restriction of the free rotation of this CN bond. Okay, now the CN bond is sufficiently the CN bond the double bond character is increased sufficiently to restrict the rotation at the room temperature. So one methyl group is cis to the carbonyl oxygen and one methyl group is the trans to the oxygen, and the anisotropy of the carbonyl group is sufficient to influence the chemical shift position of the cis group, and using the heated probe instrument. Okay, and so that is why at room temperature there are two signals, because this due to this restricted rotation. But when high temperature is used, okay, using a heated probe in the NMR instrument, the spectrum of DMF can be recorded at a high temperature around 130 degree, and the spectrum will show only one signal of the methyl group, because at the elevated temperature the rotation of the CN bond is so rapid that each methyl group experiences the same time averaged environment. Okay, so in room temperature, due to this double bond character, this resonance, the methyl group, two methyl proton, two methyl NMR signal are recorded. But when the high temperature is there, the rotation is so rapid, there is no resonance, and the only purely the CN bond is purely single in character, and the methyl groups, they show a time averaged, they experience a time averaged environment and show only one NMR signal. Okay. Now problem number 19, it says the hydrogens which are most likely to get exchanged with the deuterium when the following molecule is dissolved in D2O. So this is a molecule, when you dissolve this molecule in D2O, which hydrogens are most likely to get exchanged with the deuterium. Your options are number A, HA only, number B is the HB and HD, number C is the HB and HC, and number D is the HD only. So when this following molecule is dissolved in D2O, which of the following proton is likely to get exchanged with deuterium? When this molecule will be dissolved in D2O, which protons will get exchanged with deuterium? Let me check the chat box because we don't have much time and one more question is left till now. Okay, just let me check if there are any answers. Okay, from pharmaceutical technology, yeah, you will be eligible for this course because pharmaceutical technology will also need the particular medicinal chemistry. Okay, so I have A, B, and D, three different options for the problem number 19. Okay, since we don't have much time, so I will just show you the answer and I have to discuss this thing. So the correct answer will be HB, number C, HB and HC. These protons will be get exchanged with the deuterium when this particular molecule will be dissolved in the D2O, because this particular HB and this HC are the most acidic protons. Okay, why HB is the most more acidic than HA? Because in case of HB, it is in between this nitrogen and sulfur, and okay, so that is why and this is that is why its HB is more acidic, and HC is also only the OH, hydroxyl OH, so it is more acidic rather than HA and HD. So deuterium exchange peaks that are due to OH and NH proton can be identified by the exchange by the deuterium. Deuterium is not observed in proton NMR. Shaking the sample containing exchangeable proton with D2O causes the exchangeable proton to be replaced by deuterium, and as a result, the signal for the exchangeable proton disappears from the spectrum. So for example, this one, here is a OH peak here, but when this particular OH, this compound is shaked in the D2O, the OD will form and no peak of OH will be observed. So one way to test if the peak is from an OH is to adding a drop of the deuterated solvent like D2 or MEOD. Because the exchange of the deuterium, the peak disappears since the deuterium is NMR inactive at the proton's frequency. Okay. So now this is the principle of the exchange in case of the NMR spectroscopy. Now the last question of today's session, which says what is the order of the reaction for which the half-life does not depends on the concentration of the substrate. Your options are like this: number A is 0, number B is first, number C is the second, and number D is the third. So what is the order of the reaction for which the half-life does not depend on the concentration of the substrate? What is the order of the reaction for which the half-life does not depends on the concentration of the substrate? Your options are number A 0, number B first, number C second, and number D third. Let me check the chat box. Okay, I have answers. Number B, number B, number A, number A, number A. So number A and number B are the given answers. Let me check which of the following will be the correct answer. So the correct answer will be, it is number B, first order. For the first order reaction, the half-life does not depend on the concentration of the substrate. Okay, it's the rate constant of this. So this is the equation, rate equation. We will discuss this kind of kinetics and all the rate equation, all these things in detail in later. Just I have thought to give a brief table of about it. So for the first order reaction, this is the value of the K, 2.303 divided by T. T is the time of the reaction, which then log A by A minus X, where A is the initial concentration of the substrate and A minus X is the concentration of the substrate after the time T. So when T is the T half, means the half-life period, then A minus X will be A by 2, means at the half-life the substrate is converted to the A by 2. Okay. Then if you replace all this thing and calculate, then you will get the T half equal to 0.693 by K, which does not have that A or any kind of concentration parameter. Okay, that is why first order for first order reaction, this particular half-life does not depend on the concentration. And this all these are the equations of the first order, zero order, second order, zero order, second order, and first order reactions. Okay, this is the rate law. This is the integrated rate law. This is the rate constant unit. This is the linear plot of determination of K, and this is the half-life. So this is the table. How these things come, this all these things you don't need to know much detail. We have actually enzyme catalysis, and that enzyme, for this enzyme catalysis we will require some of the rate equations, but these things all in the week 2, so which we will discuss on the next class. Okay, so so what did we do today? We discussed about the basics of the medicinal chemistry of the drug discovery. Then we discussed about the acidity, basicity. We discussed about the structural determination, NMR, proteins and all. So I will share my email ID with you. Just give me a minute. So this is my email ID which I have shared in the chat box. If you have any doubt or any query regarding today's class, you can let me know through the email. I will answer your queries through the email. And somebody has asked where to find this problem solving session. So in the NPTEL, in the SWAM website, wherever when there is this particular medicinal chemistry course content announcement portion, right? You have all this thing. Okay, so here the in this left portion, when there is a particular this quiz and week 2 assignment and all these things are uploaded, there will be another tab which will be enabled soon, which is known as the problem solving session. If you click on this particular problem solving session tab, you will find the PowerPoint presentation which I have today shared with you, and also you will find the lecture video. I am recording today's session, so this recording I will upload to the YouTube, and this YouTube link along with the presentation link will be given to you. Okay, and this from there you can access both the video and also the particular PPT. This tab will be activated by this weekend, so you will find it by Saturday or Sunday. You will get this PPT and all this thing. So if you have any doubt or any query, you can ask me now, or you can let me know through the email. And yeah, somebody has asked me about the book of the medicinal chemistry. So there is a book of the medicinal chemistry, which is known as like medicinal chemistry by Graham L. Patrick. Okay. So this book you can download. The PDF is available. You can download this book. I think this is a very good book, a basic and very informative book for medicinal chemistry. So you can follow this book. And if you do not find a PDF online, you can drop an email to me. I will share the PDF with you. Okay. So today it was our first live session of the medicinal chemistry course. I hope the course will, this particular live problem solving session will help you to understand the course better, and also it will help you to solve the weekly assignments which are posted. So today we discussed all the week 1 course content, and for the next class, which will be on next Friday from 4 to 6 p.m., we will discuss the week 2 course content. Okay. So I will close the session for today. Feel free to contact me if you have any doubt or any query regarding, and try to get this book, because this book will help you to understand the course better. So I will close the session for today if there is no query. Thank you so much. I hope the class was helpful to you. You did understand few things, and if you have any doubts, let me know. Then I will cover the topics in the next class. Thank you all for joining. I will close the session for today. So see you on next Friday from this time 4 to 6 p.m. Okay, the second session will be on the week one itself, but it won't be taken by me. It will be taken by somebody else. If you want, you can join it. I will take the sessions only on Fridays. Okay, so the Friday session will be taken by me, and the Saturday session will be taken by somebody else. So you can attend the session. It's up to your choice. Okay, if you have time, you can attend the session. Okay, thank you so much. Thank you all for joining. So let's see you on the next Friday. And feel free to contact me if you have any doubt. Okay, so I'm closing the session.